This is the sort of corrections video. Collagen type 1 is one of the most abundant proteins in the body. And I forgot to mention that its structure of the triple helix was glycine attached to like an X and a Y of different things. But the glycine keeps that helical nature of like, because they're always on the inside of the helix. That's where the side chains go. So your vitamin C ties in because it deals with alpha ketoglutarate, because you need vitamin C and alpha ketoglutarate to hydroxylate your proline and your lysine. These are big fancy words. So proline and lysine, they're both amino acids. Proline's in a ring, like a 5-point ring. But that's not important right now. So vitamin C turns your amino acids into collagen. And the other thing besides your gums being less stable and your teeth being loose is your blood vessels are easily injured. Blah, blah, blah. We got a featured song called Scurvy from SpongeBob that our professor included in this. Lysyl oxidase is important. I don't quite understand that structure process yet. I'll have to get back to that. There's a lot of types of collagen, like 28 of them. Oh, OI is osteogenesis imperfecta, and that's your defect in collagen synthesis, not the other thing I said. There's 19 mutations of it, and sometimes your sclera can be blue, so like that's pretty wild. Part of your brittle bone. There's a lot of biochem involved that I need to get back into. Laminin, T structure, I mentioned. There's an alpha, beta, theta, alpha, beta, gamma heads to it. Again, various combinations of that. There's 18 identified, but 45 possible combinations. And then what I said about friction. So proteoglycans are the fibrous structural proteins. They have a hyaluronic acid core. So your little guy here is going to be hyaluronic acid as the base. And then you have your GAGs. And your GAGs are the disaccharides. But this is going to have like 100 GAGs on it, which are GAGs, I believe, are sugar-based, and they are 95% carbohydrate by weight, which makes sense because if you think of carbs, it's pretty much all carbon chains. There's a whole lot about GAGs, hyaluronic acid, and then what I said with you have your GAG, you have your trisaccharide, and then you have your serine. The order of synthesis is you just keep adding sugars onto that. He's repeating himself now, so it's not just me. So the matrix sounds really cool. It's your hyaluronic acid backbone. And then it binds, the link protein binds hyaluronic acid to proteoglycans with non-covalent bonds. Interesting. Back to chemistry, covalent, ionic, different types of bonds. So non-covalent, it could be ionic, could be something else. It also acts, so since it's a brush, it kind of acts like a filter, so it blocks out big particles but lets small particles through, which is very important in your organs, like your liver, kidney, things that kind of like filter stuff. That's a good thing to have, like a mesh. I explained the water part. degradation I didn't really get into, but it uses endocytosis, endosomes, fused with lysosomes. Lysosomes likes to break things down anyway, so if you have the suffix lyse or lysis, usually means breaking things down. Defects cause lysosomal storage disorders such as mucopolysaccharidosis. So I know he has a later slide on that because there's a lot. So MPS, mucopolysaccharidoses, is a whole bunch of diseases like Hunter, Herder, and Shea, Martolini, mucolipidosis 7, Sanfilippo A, B, and D. They all are different enzyme deficiencies, so you have different accumulated products. Integrins, I kind of forgot about this, so we'll just run through it real quick. But transmembrane protein, so that means it goes through the membrane. There's one on the outside, one on the inside. The inside one is your cytoskeleton, and your outside one is the matrix. I said it very dramatically, but the other thing is your white blood cells don't usually have integrins, but they can be induced. Like if you're injured. Okay, MMPs and TIMPs. So matrix metalloproteinases. They have zinc. They degrade all your ECM proteins, which is what I said earlier. Not to be like, I'm smart, but like, I knew that one. So it's highly regulated, a lot of processes. To control your MMP, your MMP controls your TIMP, your tissue inhibitors of metalloproteinases expression. So they kind of like counteract each other and balance it out. Oh, he gives a summary at the end. Okay. Let's see what I got. Elastin uses no energy to stretch and contract. Laminin is important in basal laminae. So that's what I was saying about your connecting your dermis and epidermis. And GAGs are your carbohydrate portions of proteoglycans, important in function. Failure to degrade your GAGs leads to MPS. Okay, so your MPS is related to GAGs. Fibronectin attaches matrix proteins to integrins on cells. I missed that part, but okay. Like I got the integrins bit. There's matrices. I skipped the fibronectin somewhere. And MMPs and TIMPs are important in remodeling matrix and preventing remodeling, which is kind of already what was covered, but alright, hopefully something was helpful, and I hope I got most of it right, so till next time.